Thank you for inviting me to give a talk about this project that we have. So I'm going to be telling you about the SWAB-SEQ project, which if you at UCLA, you know about this very well. You're actually mandated to use our technology every week. And I want to kind of just take you back to the origins of this project, which was in March and April of 2020. So if you remember, during that time, it was almost 2 years ago, this was the kind of things that were being published, like what went wrong with coronavirus testing in the US. COVID-19 testing is really sluggish. That puts us at risk. Tom Hanks, he went to Australia, where you can get COVID testing. The Utah Jazz were shamed for getting 58 tests, stepping out of line to do that. In fact, this problem was so acute that the state of California invested $1.4 billion in order to build a facility to do COVID testing, at the end of the summer. So our labs were being shut down. The faculty that were involved in this project were thinking, we actually are genomics faculty and we know how to measure RNA. So, and we have all the equipment and, all the research is being shut down. We can use that equipment to provide COVID testing. And that was really the origin of the SWAB-SEQ project. And at the time, the technology that was really being used for COVID testing is based on quantitative RT-PCR tests, which has been the gold standard. It's been widespread use for 20 years. But there's many limitations, logistics, purification, there's not enough machines, there weren't supply chains, and it's relatively expensive, which is what caused this massive shortage at the beginning. So what we did as a group, we developed a new technology, which is fundamentally different. It's a PCR test, but it's fundamentally different than the standard technology because it's based on genomic sequencing. And it also has the advantage that it doesn't require extraction or purification, which makes it very scalable. And it uses something called, and this is the basics of the technology. And so the two key ideas behind the technology is that one, it adds what's called a molecular barcode to each sample. So for every for every patient sample, they get a unique kind of a piece of DNA attached to them. So here's kind of a toy example. And this barcode is added to each sample in the first step. All the samples are then pooled together, sequenced. And after sequencing, we can tell which samples have coronavirus because we can look at the barcodes. So that's one of the key ideas. The other key idea is that we added something called a spike-in, which we added something that looks very much like coronavirus, but has 6 of its DNA positions reversed. And that allowed us to generate a more accurate test. So if you look at just kind of counts of coronavirus, it's very noisy, but if you look at kind of ratio of counts to this artificial spike-in, it's very accurate. So this turned out to be a highly accurate test. This is all in April and June of 2020. We showed that it's about 4 times more accurate than the standard qPCR test, which is widely used. And using that, we could actually use it without doing RNA purification to generate a nasal swab test that works very well in practice, and also a saliva test, which is the one in the vending machines that's very comparable to kind of state-of-the-art test. So we actually did all this work in about 2 months. We got the test done, developed. We showed proof of concept to the leadership, and we said, okay, there's this massive COVID testing shortage. We've solved it. Let's go and let's deploy it. And so, the genomic technology works, but we found that that the leadership actually told us the logistic challenges are equally as challenging, and so that kind of started what we call the bench to vending machine adventure. The great thing is that UCLA kind of believed in us and said, okay, let's see where this goes. So what does it take to build a CLIA high complexity lab? You need space, money, forms, clinical personnel, regulatory compliance. We decided to take this on. So UCLA provided us the 8th floor of South Tower. And so we have sequencers with the best view that sequencers ever have. We built the lab there. There's actually a key moment. There's a key moment where we're building the lab, we can do this. We go to the leadership and we say, look, here's the thing. All the supply chains are really short. And if you want us to, we need to buy tubes. We need to buy pipette tips. It's going to cost millions of dollars to do this. Do you want us to place the order or not, right? They thought about it and they said, okay, sure. So we bought millions of funnels, tubes. We started. This is what the lab looked like, and then just to give you a sense of kind of what happened, I want to kind of organize this as a timeline. So this is an example of a technology that was really developed in April of 2020, and it was in May we moved to the new space. In end of June, beginning of July, we submitted our FDA EUA application. We started with our automated processing. We designed our collection kit for saliva. We did an operational pilot with the Student Health Center in July. We bought all these supplies, hired lab staff in September. We got our FDA EUA and our CLIA license granted in October. And then in November, we were deployed to UC Santa Barbara, was our first client, and we were up and running and testing their campus. So it's basically 6 months from initiation of the idea to deployment. And we also uniquely had a situation where we were very under-resourced. We didn't really have any staff, so we had to innovate in how to make things very efficient. So, for example, when people receive samples, there's oftentimes a lot of labor-intensive components of dealing with the samples, getting them organized. We actually decided we use kind of a system to register the samples. It's the one that uses the vending machines. Samples are sent to us in racks that then get inactivated in a water bath and get onto our kind of automation machinery to process. So the whole system runs with very few staff. We also had to develop a way for how to do collection. And so one of our team members, actually developers of the technology, Valerie Albreletta, she had the idea that, instead of it was very hard to get these saliva collection devices, she had this idea that we can buy perfume funnels. And she actually found on Amazon a perfume funnel that fit exactly into the tube, and we bought 20,000 of them, and that was really what we used to get started. We then had them manufactured. Similarly, another thing that we had a lot of pressure to develop a swab test as well. But one of the most labor-intensive components of COVID testing is actually dealing with the swab, taking it out of the sample. You have to do it in a clean room, or not a clean room, but it's a biosafety cabinet. It's actually also the most dangerous part of the test. We came up with a completely different system. We actually developed with Gabe Oland, who's a biodesign fellow at the time. We actually came up with a new swab that would break exactly at a point, so it would fall into our 1 ml tube. It's actually the smallest swab, that fits into the smallest tube, and then our automation would work kind of above at the liquid above the swab. So we designed a 3D print swab, and we ended up getting this manufactured. So overall, there's many kind of efficient processing ideas that the samples were kind of registered at the collection site. We use this software from Precise MDX to be able to scan the tubes and process the system. The samples arrive in our lab ready for automation, and everything uses the same tubes. We developed all this, and because it was designed to be low staff, everything turned out to be highly scalable. So this is kind of what, once we deployed, we were testing for UC Santa Barbara. We then quickly started testing for UCLA Health and Caltech. We were testing for UCI and Pepperdine. We started sequencing COVID genomes. We started for Cal States. Then we were evaluated by this NIH RADx program that supports new technologies in COVID diagnostics. And we actually got that. And in June of 2021, we took over the UCLA campus testing With vending machines, we also started testing for LAUSD. So actually, at the time when we took over the campus vending machines, the thought was like the vending machines made a lot of sense because there wasn't going to be a lot of testing. We would be doing maybe 3,000 tests a week. But then what happened was that the Delta variant came, and we very quickly had to scale up to doing 40,000 tests a week. And then around beginning of November, things started to die down, and it looked like everything would return to normal. And at this point, we had done 400,000 tests. But then, of course, Omicron hit, and now we're doing like more than 50,000 tests a week. And we've actually very proud to say that we've actually crossed our 1 million of total tests completed. So it's the first time we've done that. So, as the tests are in vending machines all over campus. So it's why we call it bench to vending machine. And one of the really cool things that happened is that we got a $13 million contract from NIH to expand our capacity, which when they gave it to us in June, we thought, okay, like this is an example of when you make the investment after it's needed. But little do we know, it actually turned out to be very important. And using it, we were able to purchase very high throughput robotics equipment and sequencers, which actually I'm going to show a kind of a slide of it at the end. So this is our lab staff. You can see it's a relatively small team. We're processing 10,000 samples, probably have double that capacity with maybe only like 5-6 staff working there. And the other thing that I think this project really shows is that a relatively small group of faculty and students, if you have a really good idea, UCLA is the kind of environment that you can actually come up with something and deploy it. So if more information, our paper is now published, and a question I always get is kind of what's next. So our original goal when we started this project was really to develop an accurate, scalable $10 PCR test, and I believe we accomplished that. I mean, our test now is $20, but at scale, I think it really would be $10. And so our next steps are really thinking about how we can use this technology for the next pandemic. So, adding capability to identify other respiratory viruses, sequence other respiratory viruses, scale, detect unknown viruses, and then also if we find a threat, very quickly create rapid diagnostic ability for new respiratory viruses. So we believe we're very well positioned for this, for this technology. We believe that we're in a position to be what California wishes that they had, 2 years ago. And just to give you a sense of kind of what's going on in the lab, this is showing one of our kind of robots, and this is actually how our test works with robotics kind of sped up. So you can see that the whole system is now automated. We're kind of moving from partially automated now to this system, and this should be actually kind of running the test within the next few weeks. So, okay, thank you. Happy to answer any questions. This is 8 times. So this system can prepare about 1,500 samples per hour.
Hi, quick question on the saliva specimens. I've heard that, especially with Omicron, the saliva, depending on what time within the infection you take the specimen, you get positives and you get some false negatives. Did you have any experience with that?
I mean, so we did a lot of saliva testing during Omicron, and we saw a lot of positives right during that time. I know that people have said that. I think it's you would need to really get paired samples, and I don't know if anyone's really looked at that very carefully. I think that, if you have sensitive tests, you will be able to catch it. It's just a matter of like the sensitivity.
All right, thank you. Thank you so much, Eleazar. That was just an amazing journey. I love this story so much.